Hello, everyone, and welcome to today's webinar. I'm Iris Kobatsky, Associate Science Editor for The Scientist, and I will be moderating our discussion. Today, Brian He will discuss training EVO2, a genomic foundation model, on 9.3 trillion nucleotides from over 128,000 genomes, as well as the model's capabilities in sequence optimization and zero-shot sequence generation. Before we begin, I'd like to thank Twist Bioscience for sponsoring today's webinar. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point, and our speaker will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsor has provided us with some helpful resources related to today's webinar, and we've posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you have minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This webinar will be archived on the Scientist website, and we'll send you the link by email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our speaker. Brian He is an assistant professor of chemical engineering at Stanford University, the Dieter Schwartz Foundation Stanford Data Science Faculty Fellow, and an innovation investigator at Arc Institute. He supervises the Laboratory of Evolutionary Design, which conducts research at the intersection of biology and AI. He's the co-creator of EVO2, the largest AI model for biology ever built, which empowers machines to read, write, and reason in the language of life. He was previously a Stanford Science Fellow in the Stanford University School of Medicine and a researcher at Meta AI. He completed his PhD at the Massachusetts Institute of Technology's Computer Science and Artificial Intelligence Laboratory and was an undergraduate at Stanford University. So let's make sure our slides are up and running. Perfect. Brian, the floor is yours.
Great. Thank you for the introduction. Hopefully you can hear me and see my slides. Let me know if you can't. And I'm excited to be sharing the work we've been doing in training biological foundation models across all domains of life and that scale from small molecules to, or from small biological molecules all the way to whole genomes. And to begin, I wanted to start with this quote by Frances Arnold, and this is actually from her Nobel lecture, where she says, "Today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it." And so the idea is that there's been tremendous advances in DNA sequencing, that almost like genome read. There's also been tremendous advances in DNA synthesis and assembly. For example, Twist Bioscience is a company that does this a lot. This is an example of DNA write. And also genome editing using tools like CRISPR. This is DNA editing. But it's still very hard for us to compose new sequences, especially DNA sequences that are substantially different from anything that we find in nature. And fundamentally, this is because biology uses basically a foreign language that's completely unintuitive to us as humans. So here is a cartoon of what's often referred to as the central dogma of molecular biology, where DNA encodes for information that then flows to RNA, and then that information flows to protein. And at each step in this code, the sequences are completely unintuitive to us. So I probably still couldn't tell you the difference between a completely random string of ACGT and the DNA sequence that comes from an actual organism. But actually there is information embedded in these sequences. There is non-random, there are non-random patterns in this data. How do we extract it using, say, computational tools? Well, there's been tremendous advances in machine learning methods that can learn higher order patterns from raw sequences alone. And these are algorithms that are used to train large language models like ChatGPT, for example. And these models are fundamentally trained with very simple objectives, like predicting the next token in a sequence of tokens, where a token could be an English word, or a token could be a DNA base pair. And the idea is that even though next token prediction seems like a very simple task, actually, for example, in language, next word prediction encompasses many very complex tasks within it. So for example, a good next word predictor in English would have to predict or would have to learn some aspect of grammar. So for example, in this first sentence, in my free time, I like to code could be a valid completion, but in my free time, I like to banana would not be. The idea here is you need a verb in this position and not a noun. But this task also teaches a model more than just grammar. So the model would have to learn maybe some notion of semantics. In the next sentence, I went to the store to buy papaya, dragon fruit, and durian could be a valid completion, but squirrel would not be. Even though a squirrel is a noun, it's probably not something you could buy at a store, for example. But also, this task would teach a model facts about the world, or how words are associated with human sentiment, or translation between languages, or spatial reasoning, or even math. Potentially, many different tasks are encompassed in next word prediction. And fundamentally, this is because the world has left its imprint on language. So even though these models are not learning an explicit world model, the world has left some kind of imprint on the text on the internet that's used to train these models. So what about in biology? So in biology, we often like to think of two domains, I guess. There's genotype and phenotype, and genotype is DNA sequence, and phenotypes are the traits and the physical attributes of biology. And it's often these phenotypes that we want to understand. And also, these genotypes encode for these phenotypes via some complex biological mechanism. But also in biology, it's often very hard to collect data at the level of mechanism or at the level of phenotypes. And beyond very simple functions, it's definitely very hard to collect data at the level of phenotypes for large-scale machine learning efforts. In contrast, it's very easy to collect data at the level of genotypes because of advances in DNA reading and DNA sequencing. So potentially, we might be able to train a model on genotypes alone to learn information about phenotypes. And fundamentally, this is because, even though evolution acts on phenotypes directly, the outcomes of this selection are written back into genotype information via, natural selection. So in the same way that the world has left its imprint on text from the internet, the world has also left its imprint on sequence variation in biology. So potentially, a good next base pair predictor for DNA could learn which mutations are more correlated with health versus disease, or which mutations are more rare or common in populations. A good next base pair predictor could also learn how these bases come together to make codons and how these codons encode for protein sequences, and also therefore learn some aspect of protein biochemistry. It could also learn how RNA bases pair with each other to learn about RNA structure, or even how genes come together to make bacterial operons or higher level biological systems. So this is a very intriguing, appealing hypothesis. If it were true, you could potentially teach a model about diverse aspects of biology with a very simple objective of just predicting the next base pair in the sequence. But is this actually a productive route? Like, is this actually a good thing to do? So a first step that many in the machine learning for biology community have taken is, including myself, is training language models not on sequences of DNA, but on sequences of amino acids that make up proteins. And proteins are a string of amino acids from a vocabulary of 20 amino acids. And the idea is you train a language model on these amino acid sequences, where you train the model with simple objectives like predicting the next amino acid in a sequence of amino acids, or a related task, which is a fill in the blanks task, where you hide some positions in an amino acid sequence and you predict the value of the missing amino acids or the hidden amino acids using the observed unhidden amino acids in the sequence context. And the idea behind these language models is similar to the motivation we just talked about, is that if you train these protein language models on hundreds of millions of protein sequences spanning all observed evolution, then there's information embedded in these sequences of how evolution has put amino acids together to form protein sequences that could then reflect downstream functional or structural properties of these proteins. And I won't go too much into protein language models in this talk, but suffice to say that these models have been tremendously useful in protein biology, and they do learn evolutionary rules. So for example, how proteins can evolve over past evolutionary trajectories. These models also learn an image of protein structure. So if two residues at two positions in a protein are in structural contact in the three-dimensional structure of the protein, if you mutate one position, you often need a compensatory mutation in the other position. So if you see covariance or coevolution between two residues on a protein, this can give you some information about protein structure, and these language models can therefore learn some image of protein structure from sequence alone. And then you can use these rules to help guide the artificial evolution of proteins in the laboratory. For example, selecting a library for directed evolution, which we did for evolving therapeutic antibodies to have higher affinity. And more broadly, machine learning has really revolutionized the study of molecules. So for example, AlphaFold is a model for molecular structure prediction, and together with models for de novo protein design, these tools, these machine learning tools, won the Nobel Prize in chemistry last year. And this is very well deserved. Molecules are very complicated, and we're still very interested in molecules. But a lot of the motivation for this research is we're also interested in how these molecules come together to make complete organisms. But when you think about it, even a very simple single-celled organism is orders of magnitude more complicated than any individual molecule. And we want to understand biology at this level of complexity without losing single molecule resolution. And this seems very hard. It's very complicated. How do we even do this? How do we even collect enough training data to learn information at this scale? Well, fortunately, over the past 20 or even 30 years now, people have been sequencing the complete genomes of millions of organisms and depositing these genome sequences into publicly available databases. And in theory, these whole genome sequences do encode the information needed to create complete organisms, but they're kind of buried in these vast online databases and just raw text. We now need to be able to extract this information from this raw unstructured data. So this motivated a model that we put out last year, which we called EVO, or EVO1 for the purposes of this talk. And this is a DNA language model. And the idea is very simple. All that this model is doing is it's predicting the likelihood of the next base pair in a sequence. So you give it a DNA base, you give it a DNA sequence, and it just predicts the likelihood of the next base pair. So very simple setup. The model for doing this is actually, though, very large and very complicated. It's a large neural network. In this case, this neural network has 7 billion parameters. The number of parameters is related to the number of artificial neurons in the neural network. And the model is also trained on 300 billion base pairs spanning prokaryotic biology. So prokaryotes are organisms like bacteria and archaea. We also train this model on bacteriophage genomes as well. And we'll get into why training models on lots of data with large numbers of parameters is useful in a moment. But another thing we did with this EVO model is we trained it only on raw DNA sequence, and we didn't train it on anything else. But the idea is that because of basically the beauty of the central dogma in biology, the same sequence at the level of DNA directly encodes for RNA, and this RNA also directly encodes for proteins, especially in prokaryotic genomes. So even though we're training a model only on DNA, we also could potentially get RNA language modeling and protein language modeling for free out of the model. But also we train EVO in the first generation of the model. We trained it to process an input sequence of 131,000 base pairs. And at that input sequence length, sometimes called the context length of the language model, we not only get information about individual genes, but also how these genes appear in the context of multi-gene systems or even complete genomes. So the reason why we wanted to train a model at very large scale, so 7 billion parameters and 300 billion DNA base pairs in this case, is that we observed a phenomenon that has also been observed in language modeling of text or image models as well. And this is something referred to in the machine learning literature as scaling or scaling laws. And it's this idea that as you increase the amount of compute used to train the model, the ability for the model to predict the next token in a sequence also gets better. So we see that here on the graph on the right side of the slide. On the x-axis is this metric called floating point operations or flops. It's basically a measurement of the amount of compute used to train the model. On the y-axis is this metric called perplexity, which is basically measuring the ability of the model to predict the next token. It's a lower perplexity is better. And so as we increase the amount of compute used to train the model, the perplexity also goes down. So the model does better at the language modeling task. So scale is one dimension with which we can improve language modeling performance. Another dimension is architecture. So for example, we use this striped hyena architecture that has favorable scaling to other architectures like the transformer, which is a common architecture for neural network language modeling training. So once we've trained this model, what can we actually do with it? So for the first generation EVO model, we really thought of two different categories of tasks. The first is prediction tasks, and the second is design tasks or generative tasks. So here is an example of a prediction task, and this is one of my favorite things we did with this original model. So here, we're going to give the model a very long sequence of DNA. In some cases, it's just the entire genome of a very simple organism. And we're going to do an interpretability experiment where we perturb or we change the input to the model and then measure the effects of this change on the output likelihoods of EVO1 in this case. And that change or that perturbation that we're going to make is we're going to insert a premature stop codon into each of the coding sequences on this long genome sequence. And these premature stop codon insertions are actually very bad. If you put them into the actual gene in the actual organism, it would make sure that that gene does not encode for protein and therefore knock out that gene. So these are very bad perturbations that we're making. And interestingly, what we see is that when we make a premature stop codon insertion into an essential gene, the EVO likelihood actually goes down by a lot on average. But when we put a premature stop codon insertion into a non-essential gene, the EVO likelihoods don't change all that much. And so actually the model has learned which genes are more tolerant of these premature stop codon insertions than others. And even though the model does not have any explicit information about which genes in the genome are essential or not essential, the model can still learn this from raw sequence data alone. And in some cases, this performance is actually quite good. So for example, in this study by P et al., they did a genome-wide CRISPR-I screen and the genome of lambda phage, and our EVO genetic perturbation experiments almost perfectly recapitulate the results of this data set as well. Well, another class of tasks you can do with these models is what we refer to as design tasks or generative tasks. So here we used EVO1 and we fine-tuned it on CRISPR-Cas sequences. These are systems that are used for genome editing. They're very complicated. They involve a very large protein. Cas9 has almost 1,400 amino acids bound to a non-coding RNA, this guide RNA, which itself is composed of two different non-coding RNAs, a tracer RNA and a CRISPR RNA. So we fine-tuned the model to, instead of, you can think of this almost like using ChatGPT to write code for you or write an email for you, but in this case, we use EVO to write DNA for us. And then we wanted to see if these DNA sequences would actually be functional. So we synthesized the DNA with Twist, and we tested it in vitro and saw that actually the protein and RNA encoded by this synthetic DNA sequence actually had comparable in vitro cleavage to SP-Cas9, which is this state-of-the-art system for genome editing. But also the model is not simply regurgitating or memorizing its training data. In this case, the model had less than 80% sequence identity to any Cas9 that we could find in nature, including in its training data. For context, this is enough sequence novelty to break any patents around Cas9. So these molecules are novel, at least in the eyes of the US Patent Office, for example. Another thing we can do with these models is prompt engineer them to generate new genes. And actually, we wanted to see if we could prompt the model to generate genes that were even more novel and sequence diverse than the Cas9 example you just saw. And so for this, we can actually use this notion that you can prompt the model with a gene and then get another gene in its output. So for example, you can prompt the model with a portion of a bacterial gene cluster like mod A and get mod B in the output, or you can prompt with mod B and get mod C in the output. So then we asked, what happens if we prompt the model to generate proteins that in nature are very diverse and are often hypothesized to emerge via de novo gene birth? In this case, we prompt the model with this anti-CRISPR associated protein to generate an anti-CRISPR protein. So this is a protein that inhibits CRISPR. And anti-CRISPRs in nature are very novel, often de novo genetic elements. So then we can again synthesize the DNA of the model-generated anti-CRISPR proteins and test them in the lab and show that they indeed inhibit CRISPR systems. So they do work in the wet lab. But also, some of these sequences are so novel that we couldn't find any natural protein that had any significant sequence similarity to these proteins. And they resemble de novo proteins designed by structure-based de novo design technologies more so than natural proteins. And I think this is evidence that EVO, when prompted in the right way and when it has the right training data, can actually be quite creative and is not simply regurgitating its training data. It can generalize quite substantially in sequence space while retaining function. We also then prompt the model to generate 100 billion base pairs of synthetic DNA sequences spanning many different putative functions. And then the craziest thing we did with the first generation EVO model is we used it to just freely generate 1 million base pairs. And we thought that these sequences would be completely unreasonable, but actually, according to a number of high-level sequence statistics, they do look quite reasonable along some metrics. So for example, the coding density on these sequences looks about right. There are proteins on these sequences with predicted secondary structure. We find tRNAs on these sequences that cover all 20 canonical amino acids. So along some metrics, these genomes look okay. Along others, they definitely don't look like real replication confident genomes. We liken this to the first versions of image generation models, where they would produce kind of realistic but blurry images. And in the same way, these models are making a step towards whole genome design, but maybe they're generating blurry genomes, so to speak. So EVO1 fundamentally was trained only on prokaryotic genomes, so very simple organisms like bacteria or archaea or phage. We left out a third domain of life called the eukaryotes, and this is the domain of life that includes plants and animals and humans. So we wanted to extend this modeling paradigm potentially to all domains of life, and in particular to tasks relevant to human genomes as well. The problem with eukaryotic genomes is that they're much longer than prokaryotic genomes. There's much more complicated genetic architecture. There's lots of sequences, lots of portions in eukaryotic genomes that are highly repetitive or non-functional. So we needed both improved modeling and improved data processing to extend the model to eukaryotes. And so this is what we did. So a few months ago, we put out this model that we call EVO2. And this is a model trained on all domains of life, everything from bacteria to plants and insects and mammals and humans. And this model also required a lot of data curation. So we had very talented bioinformaticians that were able to filter the model and focus it on functionally relevant portions of eukaryotic genomes. This model also required much more engineering and scale. So for EVO2, we extended the context length from 100,000 base pairs in EVO1 to 1 million base pairs in EVO2. So at this scale, we still retain single nucleotide resolution, so we still can understand things at the level of genes, but now we can place these genes in the context of complete bacterial genomes in some cases, or complete yeast chromosomes, or even human TADs. And we scaled this model now to 40 billion parameters with the help of NVIDIA. This actually results in, and we trained the model at 40 billion parameters up to 1 million base pair context. And we trained the model on about 9 trillion base pairs of DNA. So this makes the model, this translates into roughly 10 to the 24 floating point operations. So for context, this is the largest model by compute ever trained on biological sequence. We also did something with this model where we made it not only open weight, so we released the model weights open source, we also released the code for training the model and the code for running the model, and we released the training data. So this model is actually what's called fully open, and by among fully open models, it's also the largest that we know of fully open model across text and code and other modalities, not just DNA or biological sequence. Great. We also invested in a lot of engineering and architectural improvements to get this model to even fit on our training infra, and we were able to scale to 1 million base pair context with an updated architecture. Right. So now we have this huge model. What can it actually do? So one important task that we wanted to benchmark this model on is in predicting whether variants in human genomes could be pathogenic or disease-causing, or variants that are benign. And actually, most of us as humans have some variation in our genetic code. And most of the time, these variants are completely benign and don't do anything. But in rare cases, they do cause disease. So we hypothesized that EVO2 has been trained on DNA from thousands of living organisms. So it's identified mutational signatures that are compatible with life. So if something has high likelihood under EVO2, it could be correlated with life or with function. But if something has very low likelihood under EVO2, it could be correlated with loss of function or with disease, or incompatibility with life. So we then applied EVO2 to score the effects of these single nucleotide changes to this gene called BRCA1. BRCA1 is a gene that is often implicated in breast cancer. And interestingly, we saw that the performance of EVO2 was actually very competitive at scoring mutations to BRCA1 based on their EVO likelihood. This is actually very competitive at predicting the functionality or loss of function of the BRCA1 gene. For coding single nucleotide variants, it's competitive with task-specific models. For non-coding variants, in this case, the model is state-of-the-art among zero-shot methods. The data is not on this slide, but the model is also especially good. It's especially, and it's state-of-the-art at predicting the effects of mutations that are not single nucleotide variants, so insertions or deletions or duplications, things like that. So, and importantly, this performance is actually what's called zero-shot performance. So at no point did we give the model any explicit information about BRCA1 function, or did we even train it with any human-specific data. It was just trained on multi-species data spanning all domains of life, and it's already getting very good performance. So then we tried to introduce task-specific data to this model by supervising a model on top of the EVO2 embeddings, and show that this supervision with additional task-specific data can improve the performance of the model even more than its zero-shot performance. So we're setting up a strong foundation on top of which you can build to train very competitive and potentially state-of-the-art task-specific models. Another thing we did with this model was we tried to interpret it. So, contrary to prevailing notions of these models as black boxes, you can actually freely inspect all the parameters and try to understand how the model is actually making some of its predictions. And for this, we are inspired by this work done by a company called Anthropic on a language model called Claude. And here, this team identified an aspect of the internal representations of this model, Claude, that only fired or that only activated when it got inputs related to the Golden Gate Bridge. And actually, they identified this feature in English, but then it transferred to other languages as well, including Japanese and Korean and Russian. So inspired by this, we wanted to also interpret EVO2. The idea behind this interpretability research is that the internal representations of EVO2 do have human interpretable concepts, but they're densely laid on top of each other, or they're in some dense combination or superposition. And to get human interpretable concepts, you need to disentangle this information and train a sparse representation. So this is what we did. We trained what's called a sparse autoencoder on the EVO2 representations and inspected the features of the sparse representation to see if these features now had pure biological concepts that we could pick up. And actually, we do find these concepts. So for example, there are features learned by these sparse autoencoders that correspond to non-coding RNAs. There's also protein-level features, so a feature on alpha helices or on beta sheets. There's features on DNA. So for example, we find features related to transcription factor binding motifs. We also find features that correspond to known intron-exon boundaries in eukaryotic genes. And actually, these features were found on an SAE only trained on the human genome. So we found these features on the human genome, but then we can also see that these features transferred to a different genome, in this case to the genome of the woolly mammoth. Finally, we can also see that, similar to how Anthropic found a feature that only fired on bugs or typos in a Python script, we found a feature that fired on frame shift mutations in eukaryotic genes. So similar to how mechinterp of language models or code models can find errors in computer code, mechinterp on biological models can pick up on errors in the genetic code. Great. So finally, this model is a generative model. And we wanted to see how good this model was now, again, at this task of genome scale generation. So for example, the model actually can now generate variations of human mitochondrial genomes that have all the correct components. And when you put these components into, say, AlphaFold 3 structure prediction, the result does resemble the known structural complex. So in this case, here is an EVO-generated electron transport chain. We can also see that the model has much better naturalness and coherence in generating genome scale sequences at the level of complete bacterial genomes, definitely better than EVO1. So the coding density is a lot better, and a lot more of the proteins generated by EVO now look like real natural proteins that you would see in a real bacteria. Though there is some additional progress to go before we get to fully replication competent bacterial genome generation. But at least EVO2 is much better than EVO1. And also now the model can generate eukaryotic genome architecture, including portions of yeast chromosomes that, or full length, really, at the scale of full length yeast chromosomes with intronic structure and promoter architecture and tRNAs. So finally, the last thing we wanted to do with this model was show how it could be used to generate sequences that are very far from anything in its training data. And for this, we apply an approach called inference time guidance. So the idea is you guide the outputs of the generative model, EVO2 in this case, with a separate scoring function that can bias the results toward properties that you might want that might be more rarely sampled by the model. In this case, we wanted to use the model to design a synthetic DNA sequence where we can control a property called chromatin accessibility. So in, say, mammalian genomes, there are regions that are more or less accessible to, say, proteins that can bind and activate DNA, activate transcription. And our task in this case was to control the location and the length of chromatin accessible regions. When you do that, we have this somewhat whimsical task of being able to write Morse code in chromatin accessible regions. So because EVO2 does not have any explicit understanding or modeling of chromatin accessibility, we have to couple it with these models called Informer and Borzoi that are not generative models, but that take in a DNA sequence and can output a predicted chromatin accessibility prediction for that input DNA sequence. So we implement this kind of beam search algorithm, if you're familiar with that, where we sample 128 base pairs at a time with EVO2. Before we continue on with that sampling, we first score those chunks, those DNA sequence chunks, with Informer and Borzoi. We accept the ones that have desired chromatin accessibility. We reject the ones that have not desirable, undesirable chromatin accessibility, and then only continue generation off of the chunks that have the chromatin accessibility profile that we want. So here is an example of one design trajectory that we use the model to produce. So on the x-axis is the position into the genome. On the y-axis is the predicted chromatin accessibility. The bold yellow line is the Morse code pattern that we wanted to design. So I'll leave the actual Morse code message that we wrote as an exercise to the reader, but a narrow peak in this case would be a dot, a wide peak is a dash, and then the heterochromatin or the close chromatin are these spaces. The squiggly white line is the DNA sequence designed by the model as it goes left to right along the sequence, and you can see how the sequence that we designed conforms to the desired pattern with this kind of inference time guidance. So in general, we do think that this strategy of coupling a very capable generative model with an application-specific scoring function or even a reward function is more general than just designing epigenomic structure. For example, you could use this to design desired protein structure or even design any function of interest if you have a sequence to function model that can serve as a scoring function. Great. So that concludes most of what I wanted to talk about today in terms of the technical aspects. Hopefully, I've convinced you that this modeling paradigm is actually quite broad and general and is capable of prediction and design tasks from the scale of genes to even complete genomes. So for example, we can predict single nucleotide variant effects on BRCA1 gene function. We can predict which regions of a genome correspond to different functional annotations. We can also predict how mutations to genes affect whole organism fitness. So for example, in determining which genes are essential or non-essential to the whole organism. We can also design individual anti-CRISPR proteins. We can design these multi-gene systems consisting of DNA and RNA and protein. For example, these CRISPR-Cas RNA protein complexes. And finally, we can potentially do design at the scale of complete genome sequences. So going back to this quote by Francis Arnold from the first slide in this talk, the way we think of how this research fits in to the overall arc of biology is that there's been tremendous progress in reading, writing, and editing DNA. And now we can learn from evolution to enable the last step in this process, which is composing new DNA sequences. And together, all of these technologies, read, write, edit, and compose, can enable true biological whole genome design. Great. So that concludes the talk. I just wanted to mention that we make all of our code and lots of methodological details for these for our study publicly available in publications and preprints and on GitHub. So these are the links and the references for Evo 1. These are the links and references for Evo 2. We even include a very detailed technical report on scaling the model and all the engineering that went into training Evo 2. Finally, this is work done across a number of very talented teams spanning machine learning expertise, including scaling and architecture, computational biology expertise, including data processing and model evaluation, interpretability work, experimental biology efforts, and actually making sure that these designs work in the wet lab. This is work done across a number of institutions, including Arc Institute, NVIDIA, Stanford, and was done with a number of colleagues at ARC, including Dave Burke, Hani Gadarzi, and Patrick Soom. And I just wanted to acknowledge all of them on this slide. And finally, thank you all for your attention. I think we have some time for questions and discussion, so I'm happy to take those now.
High-throughput screening is fundamental to biomedical research, particularly for the development of protein-based therapeutics. By iteratively screening protein variants, researchers explore a protein's vast sequence space in search of a novel variant with desired, optimized properties. However, finding the right protein variant is difficult, even with the latest AI tools. Large-scale screens and training datasets for machine learning require pools of DNA, encoding thousands of defined protein variants to effectively sample the protein sequence space. There are many ways that researchers can generate their screening libraries, but current technologies leave much of the sequence space out of reach and underexplored. If your protein is longer than 300 nucleotides, your options for building screens are either very expensive, very limited, or very biased. As a result, the sequence space of most known protein sequences cannot be effectively studied in high throughput. To advance therapeutic protein development and AI-driven screening, researchers need tools to economically synthesize longer sequences at scale. At Twist, we constantly ask, how can we give more scientists access to longer synthetic DNA on a large scale? Our team used our proprietary silicon-based platform, optimized chemistry and automation to become the only company able to synthesize precisely designed pooled sequences that push the boundary beyond 300 nucleotides. This innovative new product, called multiplex gene fragments, consists of uniform pools of double-stranded DNA that are designed to your sequence specifications. In just 1 week, you can receive pooled lyophilized gene fragments, up to 500 base pairs ready for use in your high-throughput screen. With access to significantly more sequence space, you can precisely synthesize a library to generate training datasets to improve your machine learning model, or screen single-domain heavy-chain antibody variable regions and make discoveries that will lead to the next blockbuster protein therapeutic. So, what will you do with significantly more sequence space? Get started with multiplex gene fragments today.
Thank you so much, Brian. We have quite a few questions from the audience, so we'll try to get to those. We can get started. The first question is it possible to generate the whole de novo genome with EVO 1 or 2?
So we have lots of examples in the preprints and the paper. I think what we've shown already is that at a very long scale, the sequences look somewhat realistic, but there are things that are lacking, certain highly conserved genes that need to be on the genome sequence that are not there. I think a big effort that we're taking on is improving the ability to sample coherently at the scale of full genomes. So we do think that EVO 2 is one definitely big part of that pipeline. It's the foundational part. It will also need to be coupled with additional models and things done to guide its outputs at inference time. But I remain actually quite optimistic that we can do whole genome design at reasonable scales, maybe very short genomes initially, but potentially much longer and more complex genomes with time fairly soon.
Thank you. And the next question, for pathogenicity predictions, since you're using a zero-shot model, how do you expect this to perform for genes or regions with less homology or conservation across species? In other words, how well does the model work for understanding biological novelty?
Yeah, that's a great question. So fundamentally, what the model is doing and the information that it's using for pathogenicity prediction is the information that it's learned across genetic variation from multiple species. So if you have a completely de novo gene in humans that has no evolutionary resemblance to anything in evolutionary history, then the model would probably not be that useful for a de novo gene in this case. But thankfully, humans do not appear out of a vacuum. Like most of our genes, especially ones that are implicated in essential processes or in disease, do have evolutionary homologs. So in all these cases, the model gives quite powerful signal zero shot. But it is a good point that in the theoretical case where you have a completely de novo gene, and you want to understand the pathogenicity of mutations to this gene, the model would be less useful, though potentially it could transfer information from any kind of related gene, or would still know very basic things like codon usage and things like that. So probably less useful in cases where there's low homology, but even if there's some amount of homology to things in the past, the model is a very powerful signal for pathogenicity prediction.
Thank you. And the next question is, for training on human sequences, do you eliminate the repetitive sequences?
We do. So that's a great point. So the model, and I kind of glossed over this, but maybe I can put up the slide. Where is it? So the model, actually, we have 2 stages of training. So there's a pre-training stage, 8,000 base per context, and then we do a context extension stage. We call this mid-training here, where we then progressively increase the context length up to 1 million base pair context. During pre-training especially, the model is only focused mostly on regions in eukaryotic genomes around genes. So we include all exons in a gene, and we also go, I think, 5 kb into the introns, or on both ends of the exons, and then kind of stitch that genic region together. And that's all the model sees during the pre-training phase. During mid-training, our context extension, there we actually do introduce some just full raw reference genomes into the model training. But the idea is, like, these repetitive sequences are probably not going to give the model any useful information at all and just consume compute. So for most of the model training, we actually filter out these repetitive sequences.
Thank you. And we have time for one more question. Can you provide any insight on genomics annotation and/or multi-omics prompting?
Yeah, so I think I can briefly talk about my thoughts on both. I think some of this mech-interp, the interpretability work is actually quite interesting. So for example, like we do have features that emerge from the model completely unsupervised. Or I guess you can't see my slides, but it's fine. We have features that emerge from the model completely unsupervised that predict known genomic annotations. For example, which regions of the genome are involved in transcription factor binding motifs. And actually, some of these predictions are quite good. They're better than some task-specific annotation tools we ended up finding out. But an interesting question emerges, what happens if you see a feature that has also been unsupervised been learned in an unsupervised way from the model that doesn't correspond to any known genome annotation? These features are probably prime candidates for maybe new biology, or trying to find out in what biological way that these features do associate with each other. So I think that's one very interesting annotation task that you can use the model for. And maybe some of these features do also correspond to different multi-omic or epigenomic tracks as well. For example, we definitely have TF binding, but it could be interesting to look at other multi-omic correlates of these features and actually show that the model has learned from sequence alone, things that correlate with various sequence properties. But yeah, it's a good question.
Okay, thank you so much again, Brian, for joining us. We really appreciate it.
Cool, thank you for having me.
Absolutely. If anyone has any further questions, please consider reaching out to our speaker directly. Contact information is shown on the screen. And as a reminder, the webinar will be archived on the Scientist website, and you will receive an email notifying you when the on-demand webinar is available. Make sure to follow the Scientist on social media for updates on future webinars. I'd like to thank everyone who took the time to join us today, especially those of you who shared your questions and comments. On behalf of the Scientist, I'd also like to thank our speaker again, Brian He, as well as our sponsor, Twist Biosciences. Thanks, everyone. Have a great day.